Hello, I'm Alex Brolo. I'm a professor of chemistry at the University of Victoria in beautiful British Columbia, Canada. And I'd like to talk to you a little bit today about some of the research that we do in my lab. Our research is most focused on the fabrication of metallic nanostructures, such as silver and gold nanoparticles. And we make these nanostructures because you can control the color of these nanoparticles, and you can make them all the way into the visible. And of course, because you can control the colors, we can use a hyperspectral microscope to image them. So this is an image of one single silver nanoparticle, and this is the image of one single gold nanoparticle. And you can see that we can not only see the particle, but you can also record the spectrum for that particular particle. We can use these nanoparticles to image cancer cells. So here we can use one type of nanoparticle or one color to look at the membrane of the cell, and another color to look at the nucleus of the cell. And when we put them together, we can see the nanoparticle in the membrane has a certain color, and the nanoparticle in the nucleus has a different color when you look on the hyperspectral microscope. And not only that, if you come here and for each one of these points with the different types of nanoparticles, we can take a spectrum which will uniquely identify the type of nanoparticles that you have here. This allows us to do imaging without the problem of photo bleaching that you see in fluorescence spectroscopy. So we can use the nanoparticles and the hyperspectral microscope to do cancer diagnostic, for example. So here we implanted a tumor in this mouse, and the tumor is going to generate what we call circulating tumor cell that's going to come out of the primary site, and it's going to go in the mouse blood, the circulatory system here, and then it's going to end up in the lungs of the mouse. So what we can do is that we can take samples of the blood of the mouse, and then we can mixture these blood samples with the metallic nanoparticles that we fabricated, and then by looking at the hyperspectral microscope, we can see that when there is no cancer cells, we don't see too much of a contrast. But in the presence of the cancer cells, we see a very strong contrast because of the nanoparticles that are scattering a lot of the light here. Finally, it was a pleasure in this very small amount of time to tell you a little bit about my research at the University of Victoria and how we use the hyperspectral microscope. If you have any question or any further comment, you can contact me here. Okay? Thank you very much.